Thanks for joining us for another episode of Biotech and Life Sciences CEO Stories. Today, our guest is Ivan Liachko. He's the CEO of Seattle-based Phase Genomics, a company whose core technology is ultra-long genome sequencing. He's also the founder of Genome Startup Day. That organization is dedicated to helping new biotech entrepreneurs transition from academic settings into industry and entrepreneurship. Ivan was born and raised in Kyiv, Ukraine, and moved with his family to the US when he was 11. He holds a PhD from Cornell and did his postdoc at the University of Washington, where he and some colleagues invented new uses for a technique that shows how DNA is organized inside of a cell. And those can be used to readily assemble the genome of virtually any life form. In 2015, Ivan co-founded Phase Genomics with Sean Sullivan, a former Microsoft engineer and his longtime friend and Dungeons and Dragons teammate. Phase has grown to 26 employees and is known for its culture of scientific discovery and community involvement. Ivan, let's start with first things first. What inspired you to start Phase Genomics, and how kind of has your 20 plus years of genomic research influenced the company's direction?
Yeah, thanks, Tim. Well, thanks again for having me here. It's really appreciate the opportunity. You know, my history is I was a scientist my whole life for 20 years or so before founding Phase. Really just always interested in just building new things, discovering new things, stuff like that. And you know, sort of what is now towards the end of my postdoc, we came up with this technology, and we didn't come up with the original tech, but we figured out how to use it for something else, for all these new applications. And we started using it, and it was very clear that this was very powerful, and this was going to really impact the field at large. And the sort of the academic structures were confining. There were lots of things we wanted to do we weren't allowed to because we weren't funded to do it, and things like that. And we just said, like, we have to commercialize this tech. It's too good to not be released into the world. And at that point, of course, I had no idea how to run a company. I've been an academic researcher for 20 years. No one else in the department did either, really. Like nobody's ever spun off a startup. And so they were like, well, you can figure it out. Go ahead and do it. So we did. And so we spun this company off, me and Sean, my co-founder, and we've been running it ever since. But the motivation has always been to bring impact through kind of development of new genomic tools, right? So like take this new genomic technology, it exposes a new kind of information that that other technologies miss. And if you get a handle on this new kind of information, suddenly all these things become possible that previously were not possible. And that's really the motivation of phases, to exploit new sources of genomic information to try to create, new cures, new diagnostics, things like that, new tools.
Maybe you can explain the core technology and what's behind Phase's ultra long range sequencing.
Yeah, absolutely. So importantly, this is not long read sequencing. There's a lot of next generation sequencing now that's called
Right.
long read, where you're sequencing long DNA molecules. This is long range sequencing, and I'll explain the difference and what this means. So the the key principle is, when DNA is being sequenced, I often explain it as like, those old telegraph machines where like the long string of paper is coming out and it's got a Morse code thing on it, like beep, 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 right? And imagine you have one of those, but it's like, it can long enough to go around the earth or something like that. Like that's
Okay.
kind of what a chromosome is. It's just like the super long stretch. And when sequencing machines sequence DNA, a lot of people assume that you just sort of like read it all the way through, but that's not what happens. What actually happens is you take that long string and you chop it up into little pieces, and then you sequence the pieces, and then you need computers to kind of put them together. It creates kind of like a giant jigsaw puzzle. And so what our technology does is something interesting. It reads the DNA using conventional sequencing technology,
Okay.
but it captures which parts of the genome are touching which other parts of the genome, which sounds a little bit crazy at first. So imagine you take this string, this telegraph string, this long piece of paper string, and you squish it inside of like a basketball, right? So that's what's happening to DNA in the cells. You have this super long string and it's all crammed into a little sphere, the nucleus of a cell.
Okay.
And it's in this like or it's in this like three-dimensional hairball inside the cell. And what our tech does is it uses basically a chemical, like a glue. It's a cross-linker. It goes inside the cells. And when DNA molecules are inside, they're touching each other, it glues them together. And then what you're sequencing those little touch points. And so what you're essentially getting a data set out that tells you this sequence and this sequence were right next to each other physically. They were touching each other inside the cell. Now, it seems like a crazy thing to measure. Why would you want to do this? Well, originally, this technology was developed to map how a genome folds in 3 dimensions, how it's organized, because you're essentially saying like sequences that are touching each other more, are close, sequences that are touching each other less are far. And from that, you can build a 3D model of the genome. And that's actually what really got me to fall in love with the technology originally. I did not invent this. This was done by, originally Joop Dekker's lab at UMass, and then Erez Lieberman-Aden. They developed these methods to look at the three-dimensional organization of the genome. But there's other information in that we thought was extremely useful if you take that assay and sort of flip it on its head and use it for something else. So in essence, when you get one of these data sets, you know which parts of the genome are close, which parts are far.
Mm.
So that allows you to do two things. One is, let's say let's say you're trying to like build a genome for an organism that nobody's ever sequenced before. Like if you think about the old Human Genome Project, right, where like the actual sequencing is only a part of the problem. You actually have to know how to put it all together, right? Like you're trying to assemble that long string
Right.
from pieces. Like which pieces go where? I don't know, right? Um, but what if you knew how far and how close every piece was from each other? Now you would know how to put them like how to string them together, right? Because you know
Okay.
the physical distance between them. And that's essentially what the technology does. And then let's say you're looking at a cancer genome, right? And you have what's called a translocation, right? Translocation is when pieces of your genome just swap, right? They move around, they
Mhm.
swap places, right? That is super common in cancer, and it creates these like fusion genes and all sorts of other things. But you can't really tell that with regular sequencing often because the sequence isn't changing. Pieces are just moving around. And so knowing what's far and what's close allows you to reconstruct the genome, detect new, translocations, find new
Okay.
fusion. So that was sort of the idea when we first started, is to
So,
use scaffold genomes and stuff. Yeah.
so Ivan, how does it or does it? And again, I'm not a scientist here. I'm just an interested amateur. Okay. But how does it or does it differ from what is now all over the press, the concept of multi-omics, because that's about spatial relationships, right?
Right. So I mean, it's certainly part of the whole, like the essence of multiomics is to try to gather as many different pieces of information about a biological system as you can, about proteins, about RNA,
Right.
about DNA, about where all the cells are spaced out. So it's called multiomics because it's sort of like like you take all these sources of information together, and this is one of those sources. So it's not so much that it differs from it, it's a part of it, right?
That's a part
Now,
of it.
normally when people think about spatial, they're thinking about how do different cells like how are different cells arranged next to each other in space, right? So this is a little bit different. This is like how is the DNA arranged inside of a cell, inside of a genome, right? So it's a sort of a different dimension. But the core idea is similar, right? It's a unique, it's something that's very important for biology, and very difficult to capture with normal tools. And if you're trying to build a genome, you really need an information source like this in order to really reconstruct the genomes, for cancer, for plants, for animals, whatever you're doing.
When I did some research, okay, what I found was that it was kind of accomplished via sort of an iterative kind of approach, right? Um, it, it, does that make it? Is that what that approach is? What makes it more effective than traditional?
Well, it certainly helps. I mean, a lot of computational tools are iterative because you kind of you build something at one level, then you build another level, you build another, you keep zoning in on the final outcome. What really makes this special is that it gives you this super long range information. You're capturing DNA, touching each other from all any as like the entire genome can come together. And so it's really that ultra long range distance information that you're capturing. That's more important than which algorithms get used because there's lots of different ways to approach the problem computationally, right? But again, just think of that like, you have these pieces of sequence, pieces of paper, and you just want to know how far and how close every one is so that you can put the jigsaw puzzle, you can put this thing together.
Okay, okay. Now, one sort of keyword that I ran across over and over again, which was kind of, I think, the basis for some of your earlier work, was something called scaffolding.
Mhm.
What is that, Ivan? What is it? Why is it important?
Yeah, scaffolding is this process of taking pieces of genomes and putting them together into chromosomes. It's a little
Okay.
bit less relevant these days. It's something that was our original sort of like when we built Phase, we started by scaffolding genomes for people. So researchers, they're trying to build genomes, they're trying to do like the Human Genome Project, but maybe it's for a cow, or maybe it's for corn, or maybe it's for some other organism that's important for whatever reason, but you don't have, $1 billion to do what people did in the Human Genome Project days. And so, and scaffolding, it's almost like um, I don't want to mix too many metaphors here. It's a little bit like, a jigsaw puzzle analogy where the genome is, it's a jigsaw puzzle. There's all these pieces, and if you know which pieces are close or far, you know how to put the whole thing together. That's the scaffolding portion of it. So building a genome is a process, everything from sequencing to assembly, then comes scaffolding, and there's lots of other steps. But it's a little bit, things are changing, new technologies kind of making scaffolding either much easier or even in some cases unnecessary. Now there's been a lot of these telomere to telomere genome projects where people can actually just brute force sequence a genome from end to end, so you don't have to deal with the pieces, things like that. But it was very important back when we started, and there was really no other way to do it with, or extremely, extremely difficult to do it back when we started this whole thing and back in 2015, 16.
So the point is that it was labor intensive, it sounds like, right?
It was very labor intensive. It was also computationally very expensive, and it required a lot of like esoteric sort of like lore knowledge, right? And we really wanted to flip that to make it an easy process. And now, people do it all the time.
So
Um,
now the technology does this at scale.
yeah, it's vastly easier than it used to be. We're up to something where I think we're getting close to 300 papers now that we've published. And so it's something that's really become kind of a norm in the field. That was one of my pride points in developing technology and sort of commercializing it is that going from something nobody's ever heard of to it's just an obvious thing everybody does. That's
And
very rewarding to a researcher.
and you mentioned that there are many algorithms that can accomplish um, the proximity ligation. How do you determine which one to use?
It's a there. This is a very common phenomenon in all computational biology. People just try all of them. I mean,
So
we
it's
we
trial and
we
error.
have our own, but scientists or inventors, right? They're all writing their own algorithms to do everything. They're all, baking things off. They're kind of making their own solutions and things like that. So I think people just mostly do it by, kind of like trial and error, whichever one they like best, that kind of stuff.
So now is part of the business acting as kind of a CRO for other companies?
A little sometimes, right? So basically, the business started out as, a very kind of, I think, typical startup story, like two guys in an office, like with $10,000 worth of eBay lab equipment and a little computer, basically doing services for people. And that's how we bootstrapped the company by, we came out with this tech that really was solving a problem nobody could solve. There was like this Nature paper that came out early on where we assembled this amazing genome and everybody started doing it. Um, and then we boxed it. We started making products. So then we said, instead of us doing everything, now you can have this in a box. We launched it, and now the customer could do it themselves. They would often still come to us for the computational work because that's very esoteric and hard for somebody to learn. And so we still do a bunch of that now. We sell reagent kits for customers to do it themselves. We do services where people, like a CRO, like what you said, people just send us like some leaves or something, and we build a genome for them. So that's a fairly well established part of the business. Now, what we've done since then is that was kind of how we started. And then we just kept using this technology to build new tools. Like I said, like the central model is we have a way of exposing new kind of genomic information. And so you can build new tools out of it, like you can do new things with it. So building genomes is one. That was the first thing we started.
Okay.
Then we built this microbiome discovery platform off of the same technology. And then we built this cancer sort of cytogenetic platform for analyzing cancer genomes and discovering all these genomic rearrangements that other technologies miss. And so that is a little bit more like a sort of a diagnostic sort of direction. So we're moving now a little bit more out of just kind of research services and research tools, more into developing sort of clinical things. We're moving in sort of more of a diagnostic direction on the cancer side, a little bit more of a therapeutic direction on the microbial side.
Okay. Okay. And the cancer thing is all about personalized
Yep.
medicine, right?
It sort of is. It's just the fact that, if you get, leukemia or something, you will go to a hospital, they're going to take your samples, your blood, and they're going to subject them to a battery of tests. And the reason why it's a battery of tests and not one test is because every one of those tests misses something. And a bunch of those tests are called cytogenetics. They're kind of under an umbrella of cytogenetics. It's looking at like your chromosomes swapping around and rearranging. And you need a number of different assays to test it. And our technology basically covers all of them. And so we're moving into the space with this tech that allows you to basically detect all these cryptic gene fusions and rearrangements and all the other stuff that you normally can only see through a combination of many tests. And so it's a faster, cheaper, better sort of,
Right.
better
So
mousetrap.
the economics for the oncologist are better.
Well, it's not only economics for the oncologist, it's also the timing, right? It's also the fact that, so yeah, so the economics come from scalability and just it's just cheaper to do than the other tests. But you also, a lot of times they have to wait for multiple tests to be done in one after the other. And so if you can do them all at once, it's much faster. So you also save time for the patient, you know, um, for whoever is looking at these things. Now, I should say these are all research tools. We're not doing any actual clinical patient treatment stuff. This is not a diagnostic. We are moving in that direction, of course, because that's kind of, we think that's the obvious application of this
Right.
technology. But the other thing is we discover things that other tests miss completely. And so we have a lot of data on this now. We've published a couple of papers recently on this. So in addition to just being cheaper and faster, it's also much higher resolution, and it also discovers things that other tests miss completely. And so it also provides value in that sort of in that extent.
So do you think it would have value in terms of generating targets that never were before?
Absolutely. Absolutely. Right. So there's sort of two sides to the story. One is we're better at finding known targets. So we get these clinical data sets that have already been analyzed and tested. And then we find, you know, cancer causing drivers, driver fusions and things like that, that the other tests miss. So one is it's a better mousetrap from detecting what's known. But the other angle is that we detect things that we find things that nobody has found before, right? There's a lot of these rearrangements that are just extremely difficult to catch by existing methods because of the nature of them. They might have a lot of sequence repeats in them or something like that, where it's very hard to catch it by sequencing. You miss it basically by every test except for ours. So there's also a big discovery element to this. Um, and and again, we find novel rearrangements and fusions in cancer samples all the time. And it's really a question of using this technology on a larger scale to find what's recurrent, what's happening in lots of patients, and then see if you can drug it.
So it could be both diagnostic and drug discovery.
Absolutely. Yeah, target discovery for sure. We think about we have projects going on in both directions.
And so you're doing those projects for others, is that right or not?
We are doing it for others. Um, but obviously we use that as a jumping off point to start doing it for ourselves.
Okay, okay. So some other applications, I know that you're working with a $1.5 million grant from the Gates Foundation
Hmm.
to reduce methane emissions from cattle. And I was shocked by the magnitude of those emissions. I knew that they were important, but maybe just broadly kind of tell us about the problem in terms of
Sure,
climate
sure.
change.
And this is the part where I'm going to context switch and I'm going to confuse half your audience because we're going to start talking about a completely other application of this technology that I
Right.
barely mentioned at all.
But it's kind of neat.
It's a cool thing. It really has to do with the microbial world, right? Like before this, we talked about human, we talked about cancer,
Right.
but the microbial world is kind of incredible. The particular problem that you're describing is that cows when they eat grass, they produce methane and they burp it out. And that's something like 16% of all the energy that goes into a cow comes out as methane. And methane is extremely like it's like CO2
Right. It's
for
not good.
climate change, but way more powerful. So I think one molecule of methane is the same as like 30 molecules of CO2. And cows are pumping this out all the time. And so in fact, there's 1.5 billion cows on Earth. Each one produces, I think, like 200 pounds of methane um, in a day. I might be screwing up my numbers a little bit here, but when you add it all up, like it's really a major contributor globally speaking to the climate, the global warming. We don't think about it because we see cows, but what you don't realize is if you can knock down how much methane cows produce by like 50%, that's the same as taking almost every car off the road in the
Yeah.
US in terms of
That was
climate
the
change. Yeah,
statistic you pointed out today.
they have like, they're saying it's cow emissions in the US, I think, are like 4 times the size of the airline industry, like the air travel. Like it's huge. You don't think about it because they're just cows, they're walking around, they're munching grass, right? But but it's a big problem. So we have this super cool project that's going on. So maybe I'll back up a little bit and kind of set up the stage. But remember, the whole point of what we do is we exploit new genomic information to build new things. And we have this unique hook into this genomic information. So we used our technology in the microbiome space to discover large numbers, like millions, of proteins that kill specific bacteria. Um, and we are now using those proteins to kill the bacteria that make methane in cows. So there's a protein, it's naturally occurring. It comes from a bacteriophage. It's a virus that kills bacteria, and they know how to, they have special proteins that do the killing. And so if you just take that protein and you feed a bunch of it to a cow, again, you find the one that kills the methane producing bacteria. You feed that protein to a cow, it's going to kill off a bunch of those methane producers, and the cow produces less methane. And so the idea is, if you can take that technology and scale it, you can actually do two things. You can reduce how much methane gets burped out by cows, but potentially also, because that methane is energy and it's carbon, right? If the cows aren't burping it out, they are integrating it into their, like, meat and stuff like that. So you can actually solve two problems. You can simultaneously reduce carbon emissions, and also you can potentially make, farming more efficient for the farmer by increasing sort of the energy gain, the like the meat gain of cows. And so that's what we're working on. We're working on, Gates Foundation has funded this work. We have a little bit of other stuff that I can't talk about in that space that we're super excited about. Has to do with how to deliver scalable amounts of these proteins to a billion cows. um, and
Are there a billion cows?
but but yeah, there's like, and there's 1.5 billion cows on Earth, and like, how do you deliver that? So we have an interesting solution that we're working on. Um, but it just kind of shows you, again, the topics that I'm describing, they seem like super unrelated, right? Like, there's cancer diagnostics, there's cows burping methane. We have a bunch of these proteins to kill infectious disease agents, like just like you would with the cow thing, like apply that to like some of the infectious diseases. Um, but it all stems from the same thing. And that's, I think, what's really fascinating, right? Here we're assembling genomes for plants and animals. Here we're detecting new cancer fusion rearrangements in the cancer genomes. Here we're like killing methane producing bacteria in cows, right? Like, but it
Right.
all comes from the same place. It all comes from this little trick where you're catching which DNA sequences are touching each other. Like, you never would have thought, like it all comes from that. And I think that's the power of um, innovation, capturing new information types, like, um, that's what's really exciting about research, is that you don't know what's going to happen. Like Einstein's like relativity theory leads to lasers, and we use them to scan like barcodes at supermarkets. Like who would have thought? And it's the same thing here. It's but if you capture new information, new technology, suddenly all these crazy new applications, you can suddenly do things that you never imagined were possible before. That's what's really fun about being in the space.
So with all these disparate uses, what you really have is a platform, right?
Oh yeah, for sure.
That's the most valuable thing in biotechnology
Well, some would
because
say
it's so
that.
useful in so many different ways.
In principle, yes. But
So
it's
what's
not
the
always
business plan to realize that value?
right, exactly. And so trying to make the business plan to realize that value is a little bit counterintuitive, because in theory, yes, you would say, sure, the most valuable thing is having a platform, but actually the most valuable thing is having a drug. And if you talk to investors, focus is the key to commercial success. And what I just described to you is arguably the most unfocused thing you ever,
Unfocused,
right? So
but
yes,
it's replicable. Once you do
so we're
one, you
doing a
can do
lot
more.
of, yeah, we're doing a lot of focusing on therapeutic for a long time we lifted the company up. We built the company by being unfocused, by being like trying all these different things. And now we're focusing on this pharmaceutical, on this therapeutic angle, on the infectious disease side. We have a diagnostic effort on the cancer side. But yes, the problem, there is a downside to having more than one application, which is that it draws your attention in different directions.
Yeah, but so are you talking about this? I, I may mispronounce it. Campylobacter infection?
Oh, Campylobacter. Yeah, Campy is just one of the bugs. It's just like it's just another example. Like instead of a cow, Campy is a Campylobacter is a bacteria that's a very common digestive. People get infected like food
Okay.
poisoning and stuff like
So
that.
are you working on a therapy for that, or is this just another license? Or
It's basically right. So I mean, what we have is we have proteins that can kill like any bacteria that we've identified in our database. And so we're working on Campy, we're working on these greenhouse emitting microbes in cows. We are working on hospital acquired infections and a bunch of other things. And so again, it's a platform play, but it's basically targeted therapeutics against specific microbes that can be delivered in a lot of new and exciting ways that we think um, it might fix the market problem of new antibiotic development. That's what we hope, and combat the sort of the antibiotic resistance pandemic.
right? Yeah. But so it's interesting because we're talking about a business plan. And I just came from a conference in Boston, and one of the topics was anabolic resistance. And one of the conclusions was this isn't going to come from pharma. The reason it's not going to come from pharma is there's no money in it. It'll cost a lot to produce, and you won't be able to sell it for very much. So, so where are you
That's
going here?
those are exactly the three. There is this interesting problem in the infectious disease space in that there's a huge problem globally speaking. The UN is meeting right now as we
Right.
speak about antibiotic resistance. It's predicted to kill more people than cancer by the time we get, and just in general, like many
2050.
of us have had cancer treatments. Not that many people. How many of us have antibiotics? All of us, right? So from a social impact, antibiotics are super important. They're the top priorities of all these different government agencies, and pharma will never make them because it's hard to make money. And it's for exactly those three reasons you mentioned. It's expensive to commercialize. Uh, People expect low prices because we got used to the miracle drugs that they are. And then the third part is resistance. When a new drug comes to market, when a new antibiotic is up, people worry that it's going to create resistance, and so it's called antibiotic stewardship. So you try to like not give it out so that you don't get a bunch of resistant microbes to it immediately, right? And that all plays against the problem. And what's really exciting for us is that these proteins that I've been talking about, there's a couple of things. One is they're much easier to discover and sort of like develop than small molecules because there is no weird esoteric chemistry you have to do. And also we already have a very like millions of them. So you can just pluck them like low-hanging apples from a tree. So it shortens, you still have to do clinical trials and all that stuff, but it shortens the time and it reduces the risk because you have so many shots on goal and you can use some modern AI methods to improve the proteins and stuff like that because we have so many. We're working on some ways that we can deliver them vastly cheaper than people normally do. And the other cool thing about these proteins is that there's no resistance against them. So they are like resistance resistant for scientific reasons. There's a whole bunch of literature on this. It's I think a lot, it's not fantasy. And it's amazing. These proteins, they're called lysins because they lyse bacteria. You can take a bacterial culture that looks like an IPA, like cloudy, and you put some lysine in it, and in 15 minutes, it's going to look like apple juice. It just clears these
But they have
bacteria.
a very short half-life, right? I mean,
These guys don't. I mean, we have ones that will work over like for, hours and hours overnight, that kind of stuff. So, I mean,
okay.
every protein is different. But you can optimize them too, right? If you need to improve the half-life of a protein, it's much easier to tinker with proteins than it is to tinker with small molecules. So again, it all comes back to this information
Hmm.
capture thing. The more information you have, it allows you to do all these new tricks. So so yeah, you know,
Do
talk
do
about
serums in the body break them down?
sure. I mean, different proteins get broken down in different kind of they're just proteins. There's no like super special biology. So they do get broken down at some point, but they kill their targets in like seconds. And so
So then who cares? Is that
it
your
might not
point?
be a problem. Yeah, I mean, everything in the end, biology is very mystical. And so you have to like, you can make all these in vitro claims, but in the end, it only like it's all about what works in the person. It's hard to
Right.
predict. But what we do know is that people have done this a lot. There have been, there are phase 3 clinical trials on these molecules, like they work. We just have a massive repository of them. And so, and again, because of this unique data type that we've used. So Gates Foundation gave us we had another grant from them last year where we basically mapped um, we took samples from around the world, wastewater, sewage, human feed, like microbiome samples from all sorts of stuff, and we processed them through our platform, and we've aggregated this huge database of these bacteriophages. And now you just take the proteins out of them because in bacteriophages and who they target. And we now can take these lysine proteins out of their genomes and use them therapeutically. And so we're doing a lot of work in that regard, starting with cow methane bacteria, can't be other kind of
Okay.
in
Okay. Wow.
but we
So
really
there's
think of it could be it could really be a transformative in the infectious disease space. And, as a platform developer, like we're always thinking like, what is our technology most valuable for, right? We can do all these different things. It's a hammer that hits many nails. What's the best nail? And right now it looks like being able to solve the AMR crisis. It's hard to get a bigger nail than that.
it's hard to get a bigger nail.
Yeah.
Um, I hope it has a big head on the top of the nail, you know.
Yep. Yep. We
Um,
do.
so, well, one of our kind of shared interests is in helping new founders in life sciences and especially academicians in their journey from academics to entrepreneurship or an industry. And you started this group, Genome Startup Day, uh, to assist in that. Can you tell us about that?
Yeah, definitely. It's a topic that is very close to my heart. So I think I mentioned this in the beginning, but when we were spinning off Phase, we were the first spin out from our department. Now, the University of Washington Genome Sciences Department is considered one of the top in the world. Like I think it's like number 4 or 5 in the country usually.
Okay.
Um, it creates an enormous amount of genomic technology. Um, but then if that's the case, how are we the first startup, right? If this department was in Silicon Valley, there would be like hundreds of startups coming out of
Right.
it. Um, why is that? Why were we the first? And and I felt that the reason why is that people, I was an academic my whole life, right? And so then I moved over, and there is this gulf between the two groups, right? And I really felt like the one of the big reasons is the psychological barrier that if you're like a postdoc and you have some invention, you're thinking about commercializing, you don't have people to look up to. You don't, it seems like it's something that someone else does, not you. It's people in Boston, it's people in San Francisco. Like I would never do such a thing. It's not for me, right? And so you just take comfort in academia. And the kind of startup events around town that I would go to, they would be about like how to raise money and how to do your lean startup stuff and how to do, right? Like educating people how to build companies, but they weren't communicating the right information because I didn't already have a company and I need to learn how to fundraise. I need to know what's it like to be a founder, right? Like even more basic than that.
Mhm.
The reason why people in something like Silicon Valley or Cambridge will spin out companies all the time is because they see everyone around them doing it. And we just didn't have that here. And so what I wanted to do is I wanted to make an event that wasn't about educating on like the mechanics and the right way to make pitch decks and all this kind of stuff, but really to just bring people from the department who became entrepreneurs and put them in front of the people, in front of the students, in front of the postdocs, and say, here's what I did, here's what life is like. And really much more like human focused and much
Mhm.
less And so that's what we did, and was super successful. We it was originally, this was pre-COVID, so it was it was a day symposium in the Genome Sciences building where I went for many years for seminars like this. It's familiar. It's not an other. We're trying to take the otherness out of it.
Okay.
And then, COVID happened, so we moved it to virtual. And so, but the idea is always the same. There's many, if you're interested in learning how to raise money, if you're interested in learning how to build slide decks and other parts of running a company, there's a thousand resources for this. But if you just want to be like, I don't even have any questions, I just want to see a founder
Mhm.
talk about themselves, like on human terms, that I think there's an activation barrier that needs to be broken, right? And that's what we really seek to do, is to get people to communicate even before they have a company. Because what you're trying to do is, there's a million academics, a million researchers who are super, super smart. Why aren't they all spinning off companies? Well, one of the reasons is that, like, to spinning off any kind of innovation, and I'm stealing this from someone, I don't remember from who. Every time you innovate, you have to basically stand in front of a bunch of experts who are smarter than you. They've been around. You trust them, you respect them, and you have to say no to them because they will tell you not to do it in one way or another. Either they will say, like, why are you throwing your career away? Or they're going to say, like, if this was a good idea, somebody would have done it already. Like, that's
Right.
a common
That's
one,
a big one.
right? and and you have to hear that from people you trust and respect and say no. So it takes a certain amount of chaos, right? It's not enough to be a good scientist. You have to be a chaos person, because you have to break a bunch of walls and push a boulder up a hill that no like and so and we're looking for those people, and you don't know who they are, right? And so you need to start doing a lot of this networking, a lot of these. You have to, like, have all these events. You have to expose them to other founders, because you're trying to figure out, like, you're trying to nurture that spark that's going to
Mhm.
make them spin out. And it's not that they're better than other scientists. It's not. Sometimes maybe it's the opposite. But spark is what you need. That chaos spark is what you need to form a
Yep.
company. And so you're trying to provide that opportunities for people, like, to find those people. And that's really what I want to do. Like, I just wanted to have a place where people can schmooze and just talk about what it's like to be a founder. And that's what Genome Startup Day was, and is. And we've done some virtual episodes and things like that.
What a great idea, because I founders ask me, they'll ask about their deck. Really? You have to have a deck. That's true. But it really doesn't matter. No one's going to give you money because of your deck. They're going to give you money because of you. They're not scientists. They're money guys. They know some science, you know,
Yeah.
enough to be dangerous,
Yeah.
but
Yeah.
they want to know that you can roll the rock up the hill.
Right. And that's the thing, is the, like, that grit. They say, what is the number one thing of success? It's grit, and it's this mysterious thing. But a lot of it is just, like, not listening to people.
Yeah,
A lot of it is just making people mad and and saying, yeah, you're right. That does sound like a real, but I'm just gonna do it anyway. Or, like, that kind of
right,
stuff. And you
right.
still have to be good at science. You still have to have good slide decks. But I think that the slide deck thing gets addressed by everyone. And no one wants to talk about the first thing, which is, you know, you have five postdocs here. Like, that one really needs to be spitting off a company. You know what I mean? Like, that kind of thing. And let people self-select and give people opportunities to do it. Because it's not just money, it's not just science, it's personal, right? And so you have to have personal
Right.
conversations and personal engagement.
It's
So
never
that,
about money.
yeah.
It's never about money. If it's only about money, you won't have the grit long enough to roll the rock up the hill.
It's true. It's true. I
Give
mean,
up.
it's certainly, like, it's tantalizing, and people, that's all you hear in the news is, like, how much money the company sold for. No one ever talks about, like, founder X feels super satisfied because they built this cool technology. You know what I mean? That's not a headline. A headline is, you sold a company for $200 million. but so we neglect that a lot of the startup stuff happens like in the heart before the like the slide decks and stuff happen, right? So that's the thing that I really wanted to help people with.
So if you had to put it down to one sentence for an aspiring entrepreneur, what would you say? What would be your best advice?
I think my favorite piece of advice to people who are thinking about it is to just talk to everyone about it and ask questions and don't be shy. People want to talk to you about this. You're not inconveniencing them. Just say, I am hoping to learn about X, and then just have coffee with people and network with people you don't know, reach out, cold call people. So I think that's what breaks the dam is when you just start talking to lots and lots of people.
Break out of your shell.
Yeah.
Great. Ivan, I want to thank you so much for being our guest today. I really enjoyed it. Um, and I learned something. And I learned
Thanks,
something
Tim.
about founders too. Um, and I think what you're doing there has to be tremendously valuable to Seattle. And I know it would be here in Atlanta, where we're trying to generate a hub ourselves. So, um, but anyway, I'd like to follow you through your journey and thank you very much for your time.
Thanks, Tim. Thanks again for having me.
Thanks for tuning in to the Life Sciences and Biotech Podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.